Thank you, and welcome everybody. It's great to see a nearly full room, as usual, at this incredible event. Thank you to the Calgary Public Library for hosting us here and for providing such a wonderful venue in which to have a gathering like this. So, who's been to one of these before? Maybe raise your hand. So a bunch of folks have been here before. Who hasn't been to one of these before? Okay, about a 50-50 split, maybe. So welcome back, everyone who's been. Hopefully that means that you've enjoyed what you've seen previously. And to those who are here for the first time, I think you're in for a nice event. Maybe even, I hesitate to call it a treat, but I think it kind of is, actually. Why do we do these? I'm a scientist at the Arnie Charbonneau Cancer Institute. I lead the Riddell Center for Cancer Immunotherapy. And, a number of years ago, we decided that as a cancer research community, we wanted to engage a little bit better with truly our stakeholders. You are our stakeholders, the patients in this audience, the family members, the community members. We've all been impacted by cancer in a variety of ways. And this was an opportunity, and it still is an opportunity for us to have really a conversation. It's a gathering place, as was said here. We're having a conversation with you about the incredible advances that are happening in cancer research and how those advances are being translated into new treatments that are having real impact on patient lives. We take you through a little bit of the process of doing that, the research process, and how that plays out to some degree in our labs at the university and through interactions with patients, with families, with our clinical colleagues, et cetera. But really, this is meant to be a conversation. We have a gathering at the top there where there's food and drink and students and trainees. And we have a moderated session here for questions and answers and comments. And I want to, encourage everybody to view it this way. This isn't us downloading information onto you. This is an opportunity for us to share what it is we do with our primary stakeholders and for you to ask questions and to make comments and provide feedback to us. And ultimately that will shape this research process collaboratively with you most effectively. So to those who have done that previously, thank you. To those who are here for the first time and are going to do that tonight, thank you in advance as well. There is a great program here tonight. I think for those who have been here already, we've talked a lot about cancer prevention, cancer treatment, cancer research on treatment and prevention. And in particular, I think there's been quite a heavy emphasis on how the immune system can be harnessed through very sophisticated new technologies, genetic engineering, synthetic biology, to create a new class of treatment for cancer, which in many patients is starting to show real efficacy. And this is really exciting. I think anybody who knows me knows how excited I can get about this area of research. And tonight we're going to build off of that really 4-year conversation. This is our 12th Cracking the Cancer Code, which is amazing. And a number of those have been in this area. And we're going to build off of that conversation in really a couple of key ways. So the first will be a talk by Dr. Nikki Wright, who is at the Children's Hospital. I won't do a large introduction here, Christina. I don't want to steal all your thunder, but I do want to just set the stage here and just provide a little bit of context for why we have the narrative arc of these 3 speakers, or these 4 speakers here today. Nikki's going to tell you about something that is amazing, that has very little to do with cancer. She's not a cancer scientist. She's an immunologist, a hematologist at the Children's Hospital. But like us cancer scientists, she uses the same sets of incredible genetic engineering technologies to solve really difficult problems in her patients with rare diseases of their immune system. We harness the power of the immune system to treat cancer. She harnesses the power of genetic engineering to treat immune deficiency, and she'll tell you a little bit about that and some of the advances that are being made in that world. Then we've got Dr. Kyle Potts, who runs the biomanufacturing program at our research center. When someone like me has an amazing idea of how we could potentially treat a patient with a rare disease and starts to dream about the possibilities, which I encourage all my students to do, it is the first step in this process. We eventually end up having conversations with Dr. Potts and people on his team about the realities of the situation, right? At the end of the day, we do have to create real medicines for real people, not just for mice or animal models in the lab. So Kyle runs a growing research program here and a biomanufacturing program that allows folks like me to bring the ideas that I have toward real medicines for real patients that get delivered in the hospitals with our clinical colleagues. And he's going to take you under the hood a little bit about how that happens and how, importantly, how when that happens locally here or anywhere, it can enable a type of translational research that really accelerates the creation of really impactful knowledge. And then our third speakers, our two speakers, Camry Imlach, a graduate student, and David Spence, a former media personality. Sure, we'll go with that. Who some of you may recognize from television previously. And they're going to describe something really neat, which is a new program within the Charbonneau Institute that partners patients or patient family members with trainees in our institute, so that really, so that both of those, but I would say in particular, the trainee can have an influence from a real patient or their family on the types of research that they pursue and the way that they pursue that research. This is a new program that we're really proud of in the Charbonneau Institute and in the Riddell Center, and they're going to describe their experience in that program and how it has impacted both of them individually as people, as researchers, and as community members. So with that, I'm done. I will hand this over now to our moderator of 12 in a row, I think, right? Maybe 11, almost all 12, Christina Frangu.
Hey, thanks, Doug. So thanks for coming, everyone. And that was a wonderful introduction. And Doug is obviously a brilliant scientist. I'm here as a journalist. And so I see this a little differently. And when I think about how I often write about cancer research, I'm often describing to the public something like science is happening in flashes, almost like, oh, here's a breakthrough, or this is answering a clinical trial. And tonight, what we look at in these talks is, I think, the hidden chain of things that goes on in between all those flashes, because that's really not the way that science happens. And so we're going to explore different parts along that hidden chain tonight and learn about how cancer treatments are being built today. You're going to hear about science that I think is just wondrous. And I was reading an op-ed about gene editing earlier this week, and they described it as the most important medical story of the decade. And that's what we're going to dig into tonight, but not just the first discovery, but all the things that go on to making sure that it's getting to patients. And there's a wonderful local story running through this, and Doug is a big part of this. Calgary is a place where we're not just talking about what's going to happen in future, but really this team here is like a key part of planning for that. And so I'm really excited to be here. Just pay attention to like how cool all the science is, but really the path that things have to travel before that they can get to patients. And so with that, sorry, I don't usually like to switch papers in front, but anyway. Our first speaker tonight is Dr. Nicola Wright. She is a clinical associate professor of pediatrics at the University of Calgary's Cummings School of Medicine, where she holds a Barb Ibbotson Chair in Pediatric Hematology. Her work has changed the lives of kids in many ways, and one of the things I found out when I was reading about her illustrious CV is that when she came back here after her training, children with severe immune deficiencies no longer had to travel out of the province for care. That's pretty amazing. And that's been some time ago. So now she's dedicated to developing new gene therapies for children with rare blood and immune disorders. Nicola?
Thank you so much. What if I told you that treating rare disease could help all of us lead longer and healthier lives? The cancer, in some ways, can be considered a rare disease. And that breakthroughs researchers make in cancer could unlock treatments for countless other conditions. My job has been to work at the frontier. My team is focused on developing novel living medicines that will revolutionize care for rare disease. Let me tell you about baby KJ. KJ is a special little boy in Philadelphia who was born with a rare metabolic condition that causes liver failure and neurologic complications. He spent the first months of life in hospital. Standard treatments were not working very well. A team of researchers in the US got together and designed novel gene editing therapy for KJ's genetic disease, along with a nanoparticle to deliver the editor to the baby's liver cells to correct his genetic mistake. He was treated with several IV infusions and is now growing well and thriving. His therapy is historical and represents a landmark achievement in science. My team right here in Calgary aims to develop similar gene therapies for children with rare blood and immune disorders, where they can be cured with just one IV infusion. A lot of the technology I'm going to be talking about is being used to treat people with very rare diseases, like multiply relapsed cancers or children like KJ. For example, we may only expect to have 1 or 2 babies born a year in Canada with some of the diseases that we are researching. So why invest so much? It can take 5 to 10 years or $10 million or more to develop a treatment for just a handful of patients. First off, each rare disease on its own is rare, but collectively, 1 in 12 people have a rare disease. With these technologies, we're at the stage of building the runway, while also trying to fly some planes. Baby KJ was one of the first planes to fly. Over time, we will complete the runway. And then we'll be able to fly more and more varied planes. By developing these therapies for some of these rare diseases, we advance the science and can apply it to other diseases, all while the cost and time investment decrease. For example, there may be a day where we can do gene editing for more common things, such as people who carry breast cancer genes. We're already doing gene editing for people with sickle cell disease, a blood disorder, which occurs in 1 in 2,700 Albertans. By advancing the technology, we're always finding new applications. I'll discuss how CAR T-cell technology can be translated into treating other diseases than cancer, like autoimmune disease, and the synergy between CAR T-cells and gene editing technology. Most of the therapies I'm going to be talking about involve my favorite powerful little cells called T cells. T cells are central to how our immune system adapts to our environment. If you can't make T cells, you'll die from infection. There are a number of different types of T cells. Most importantly, they're the traffic cops. These are the cells that tell the immune system where to go and what to do, when to start inflammation and when to stop. And the snipers, which kill virally infected T cells and hunt and kill cancer cells. In essence, I think T cells are the bomb. That's where my kids think I'm a geek. Why this talk of T cells? You may have heard about CAR T-cell therapy for cancer. CAR T stands for chimeric antigen receptor T cells. These are T cells that have been genetically edited to target specific proteins or markers on the surface of cells. They can be made by taking a patient's own T cells or using a different donor. The donor T cells are then collected through an IV with a special apheresis machine, taken to the lab, edited, and then infused back into the patient. Those edited cells then go find the patient's cancer using the protein marker they have been programmed to recognize. When they find their target, the traffic cop CAR T cells come and secrete chemicals that activate the rest of the immune system, telling it to come fight. And the snipers essentially poke a hole in the target cell and inject toxic chemicals that kill the cell. Once activated, these CAR T cells also start dividing, making even more fighter cells. An example of these is CAR T cells genetically edited to recognize antibody-making B cells. These CAR T cells can be used to fight refractory leukemias, lymphomas, and multiple myeloma, cancers caused by antibody-making B cells that have gone rogue. Clinical trials have shown remission rates of 40 to 70%, in patients who have relapsed multiple times and have tried multiple different types of chemotherapy. Many of these patients had no other treatments available and were saved by CAR T cells. The challenge with CAR T cells is access. It costs $500,000 per patient to make them, and currently commercialized products are made in other countries, though there is work ongoing to make them in Canada. So what about CAR T cells for non-cancer diseases? Turns out many autoimmune diseases are also mediated by these antibody-making B cells. So what about trying these CAR T cells for autoimmune disease? Many of these patients do not respond to standard immune suppressants and drugs that deplete these antibody-making B cells. As a result, they suffer with chronic pain, organ complications, frequent hospitalizations, and poor quality of life. There have been a number of studies done in other countries using CAR T cells that have shown high rates of remission in patients with refractory autoimmune disease. With CAR T treatment, remission can be obtained with just one IV infusion, and it can last more than 2 years. This is compared to having to take medication daily or frequent injections or infusions at a clinic. Similar to CAR T cells for cancer, this therapy offers patients a vastly improved quality of life. As lead of the Pediatric Cellular Therapy Program, I'm excited for our team to be embarking on our own made in Canada CAR T cell trial for refractory autoimmune disease. Cells will be made in Canada. The trial, led by local researchers Drs. Kevin Hay and Tony Trong, will open at several sites across the country. With this trial, Canadians will finally have access to this novel therapy, and it will help us build the capacity in Canada to produce CAR T cells for a variety of indications. So now I'm going to talk to you about a disease you might have heard about, the bubble boy disease, severe combined immune deficiency, or SCID. Patients with this disease have no immune cells, including those powerful T cells. Without bone marrow transplant, SCID is fatal in the first 2 years of life. The first genetic therapy that was used to treat a specific type of SCID, called ADA-deficient SCID, were made similarly to CAR T cells. Researchers were trying to develop gene therapy for ADA SCID, but were initially having problems correcting enough bone marrow cells. CAR T cells were just being developed. And in 1990, researchers decided to try genetically modifying T cells instead of bone marrow cells to see if that was sufficient to correct ADA SCID. 2 patients were treated. They took the patient's T cells, infected them with a modified virus containing a functional ADA gene, and reinfused the T cells. Though it wasn't a cure, one had improvement, Ashanthi De Silva, who is now alive and well, and is a genetic counselor and an advocate for these novel treatments. So this leads me into gene therapies. The scientists were eventually able to overcome the challenges in correcting enough bone marrow stem cells to create effective gene therapies, first for ADA SCID, then other immune disorders. This is currently done by collecting the patient's bone marrow stem cells, taking them to the lab, infecting them with a modified virus containing the normal gene, then infusing them back into the patient after giving them a dose of chemotherapy. There have now been over 100 patients with ADA SCID treated with gene therapy. All are alive, and over 95% no longer have to live in a bubble. Now, you may have heard of CRISPR, a newer type of gene therapy. This technology utilizes a modified DNA repair system from bacteria to correct or gene edit human DNA mistakes. To understand genes, these are the blueprints to make your body, like the book of instructions. Each gene is like a word, and the instructions to make a protein. When a patient has a genetic disease, they have a mistake in one of these genes. Genetic mistakes can include missing all or part of the gene, a spelling mistake, or extra letters added to the gene. When we use a modified virus for gene therapy, a normal gene is randomly inserted into the patient's DNA by the virus. It's like the whole word is inserted into a random place in the book. With gene editing, The stem cells are treated with CRISPR to correct the DNA mistake. This is more like a spell checker that goes in and corrects the word right where it is in the book. There are other gene editing technologies that have been developed building off the CRISPR technology that have some advantages. Base editing utilizes a modification of CRISPR and acts as a spell checker or a pencil for a single letter mistake or point mutation in the gene. This remarkable technology has the capability of finding one mistake in over 6 billion letters, highlighting just how incredible this technology is. It's more efficient, correcting more cells, and more precise than CRISPR. I am excited to be collaborating with Dr. Don Cohen at UCLA, one of my heroes, and one of the key people who developed the ADA SCID viral gene therapy, to develop base editing therapy for another type of bubble boy disease called CD3 delta SCID. This is one of the most common types of SCID seen in Canada. In these patients, they all have the same one-letter mistake in their gene, a T where there should be a C. Once optimization of therapy is complete in the next 1 to 2 years, the clinical trial will open in Calgary, the lead site, as well as Winnipeg, Toronto, and Los Angeles. Another new technology is prime editing. This also builds off CRISPR, but can edit more than just a single letter mistake, more like a word processor. It needs more optimization. However, there was recently a clinical trial of another rare disease, an immune disorder called chronic granulomatous disease. The trial opened first in Canada, which is unusual because we're often last in these games. The first patient treated was a patient from BC treated in Montreal. This was a landmark for Canada and a noteworthy milestone, being the first patient treated with prime editing. So where are we in Calgary? I'm excited to share that we are currently developing a platform for gene editing technologies where we will be able to design personalized gene therapies for patients with inherited blood and immune disorders, just like baby KJ. The next stage in development is moving these therapies from modifying the cells outside the body, which takes a lot of time, has to be done individually for each patient, and is really expensive, to developing technologies where the gene editing machinery can be given directly to the patient. To do this, the gene editors will be packaged in delivery nanoparticles, technology we are developing locally. In the future, this means with just one IV infusion, the gene editors can be given to a patient to ideally cure their disease, whether it be to genetically modify their cells, to create CAR T cells, to fight cancer or their autoimmune disease, or to fix an inherited gene mistake causing their disease. Our team is working on this technology right here in Calgary, where this work is positioning us as leaders, both nationally and globally. We are already building the runway and plan to start flying planes in the next 1 to 2 years. These are extraordinary tools, and what we've learned can be translated into treatments for a wide spectrum of disease. There are other baby KJs out there. The progress we've made so far represents how work in rare diseases will help us develop therapies for other diseases that will impact many, many more people. As I look to the future, I envision a time when a patient, like KJ, is diagnosed with a rare disease. Our teams get to work creating a personalized gene editing therapy, and we package it in a special nanoparticle delivery device, and we are able to offer them a cure with just one short IV infusion, thereby saving lives, reducing treatment toxicity, and restoring quality of life for patients and their families. Thank you.
Thank you, Nicola. If you are a geek, We are your people. Next up, we have Dr. Kyle Potts. He is the Associate Director of Biomanufacturing at the Riddell Center for Cancer Immunotherapy at the University of Calgary. And so what he does is he's leading the effort to build and operate one of Canada's first academic facilities dedicated to manufacturing cell and gene therapies for patients. And he's going to tell us a little bit about that. Kyle?
I want to take you back 5 years inside the lab at the University of Calgary. A research lab much like the ones you've seen on TV were pictured here. Bright lights, crowded benches, pipettes, racks of tubes, boxes of gloves. That sharp, clean mix of disinfectant and cell culture. Nothing dramatic, nothing futuristic, just a working research lab, the kind of place where science happens every day. And in that room, a scientist is looking at a young woman's cancer tissue. And he sees something important, a protein on the cell surface that is present on the cancer, but largely absent from the patient's normal tissue. And he has the kind of thought that people often associate with a breakthrough. What if we could build a new therapy to this protein? Could that help this patient and others like her? And that's exactly what happened. Here in Calgary, that insight became the beginning of GCAR1, a novel CAR T cell for solid tumors. It's an engineered immune cell designed to recognize and attack that very cancer protein. But that's where I want to pause, because it's the part that's often missed. A breakthrough in the lab is only the beginning. Innovations like GCAR1 don't reach patients on discovery alone. They reach patients when discovery is matched with the ability to build, test, learn, and improve. And with recent advances in biomanufacturing, we now have the capability to do this, to make these type of therapies locally, close to the researchers who make the discovery and close to the patients who need them. And this is why local biomanufacturing infrastructure is so important. It's what allows innovations like GCAR1 to become real therapies for real patients. And this is why I believe so strongly in what we are building here in Calgary and what I want to tell you about tonight. So let's go back to that story of GCAR1. Once the discovery was made in the lab, the work kept going. The team worked to build the best version of that therapy they could. They tested it in cancer cell lines in petri dishes. They tested it again in animal models of the patient's tumor. And for GCAR1, those results were extraordinary. In the petri dishes and animal models, we wiped out the patient's cancer cells and tumors. But studying a new therapy in humans is much different and much harder. Moving to a phase 1 clinical trial means testing a therapy that has never been tested in people before. The main goal is to understand safety, but it also begins to tell us if this therapy might actually be able to help these patients. With cell and gene therapy, the treatment is not a simple chemical formula that can be manufactured the same way over and over again. In many cases, cell and gene therapy means taking cells out of a patient, engineering them and expanding them in the lab, and then giving them back to the patient as a treatment. That is not simple. It requires working with living cells from a real person. And those cells must be purified, modified, grown, tested, and returned to that person in a way that is safe, consistent, and can help fight their disease. And that's where our biomanufacturing team comes in. My name is Kyle Potts, and I lead a growing biomanufacturing program at the University of Calgary. And this is the work we do every day. Biomanufacturing operates at the space between a discovery in the lab and a therapy that can be given to a patient. We take research-grade ideas that look promising in the lab and figure out how to make a clinical-grade therapy for patients, meeting the highest standards of quality and safety along the way. In other words, this is where a scientific idea becomes a real clinical product for real people, not just cells and mice in the lab. And that's what I want to highlight here. This is the manufacturing process we use for GCAR1, but it'll be in 2 very different forms. Across the top here is the research scale workflow we use. This is how GCAR1 was first developed and tested in the lab. It's small, open, built for speed and flexibility. It lets us ask the first essential questions. Can this therapy work in preclinical models, and how do we make it better? Across the bottom is our clinical scale workflow. This is the process we use to manufacture GCAR1 for patients. It's larger, more controlled, built around closed systems, defined steps, and rigorous quality checks, all within a highly regulated environment. Only then can it be given back to a patient. Same therapy, but 2 very different processes and standards. And this is the difference between testing an idea in the lab and making it safe and consistent enough for patients. And that shift from discovery to delivery is part of what makes GCAR1 and other living medicines so different from the type of drugs we're used to. Until recently, medicine mostly meant giving a person a chemical drug, something manufactured on a large scale, hoping it would change their biology in the right way. But cell therapies are different in an important way. The therapy itself is living. It's more than a molecule. It's living cells that carry out their function inside the patient. Sometimes these are the patient's own cells, altered so they can better recognize and fight cancer. Sometimes they're donor cells as ready-to-made therapies. And sometimes there are cells carrying new genetic instructions to correct or redirect how these cells behave. And this is already happening. These therapies are already reaching patients and improving outcomes. But we want to do it better, and here in Calgary. We know that the process of making cell therapies helps shape the product itself. It matters how we grow the cells. It matters how we engineer them, handle them, and test them. As we often like to say, the process is the product. And that's why manufacturing is not just what happens after the science is done, it's part of the science itself. And for me, that is one of the most compelling parts of this field and this job. Taking something that looks promising in the lab and helping figure out a path forward towards the patients. So what happens next in a story like GCAR1? In a perfect world, it gets manufactured close to the science. We call this decentralized or point-of-care manufacturing. And this is what we are building here in Calgary. And this is critical because it keeps this learning loop tight. The researchers developing the therapy can work with the biomanufacturing team and the clinicians treating the patients, all happening close to the patients themselves. And this matters enormously because the first version of a therapy should not be the final version. Like any complex technology, it gets better through testing, learning, and improvement. A therapy may be promising in the lab, but still needs to be made at a scale that is reliable and consistent before it can be delivered to a patient. In that local model, what we learn from the early patient experiences can directly shape the next round of development. A promising idea like GCAR can move into the lab, into clinical trials, and what we learn there can quickly feed back into the next better version, GCAR2. And that's why local infrastructure matters so much to us. It gives us the chance to learn, improve, and move promising ideas from the labs to patients quickly. And over time, that can help move more patients access to these therapies. For GCAR1, that local learning loop really matters. It's not just theoretical anymore. We've already treated 2 patients here in Calgary. We now have a national phase 1 clinical trial open that would begin enrolling patients next month. And importantly, while that local infrastructure enables that learning, it doesn't just stay in Calgary. It is shared across a broader network in Canada that we are helping build. And that's why this local ecosystem is so critical. You see, breakthroughs do not move to a patient through one discovery, one clinician, or one facility acting alone. They move when biology, manufacturing, clinical research, and the patient experience work together. And that's what we are building in Calgary. Not just an individual expertise, not just one facility, but a connected ecosystem linking discovery, manufacturing, clinical care, and ultimately learning. At its core, biomanufacturing research is about taking something that works in the lab and figuring out how to make it safe and reliable for patients. Let me give you an example. In the case of GCAR1, one of the things we've worked on is reducing the amount of lentivirus used in the manufacturing process. Like you've heard, lentivirus is one of the most critical reagents for engineering these cells. It is also one of the most expensive. And if we can use less of it while still making an effective therapy, we can stretch those materials further, treat more patients, lower the cost of manufacturing. And we have developed a process to do just that. And I'm really proud of what our team has achieved. But it doesn't just end there. That knowledge, we're now sharing that with colleagues across the country so they can do the same with their processes. And that's what this work really is, learning everything we can about a discovery, optimizing every detail of the manufacturing process so the final product is safer, more consistent, more effective, and more affordable. We learn what works, what doesn't work, we learn what needs to improve, and we use that knowledge to build the next version even better. Importantly, what we're building in Calgary is not just about one project. It's a foundation for many projects like GCAR1 and what comes next. We're working on improving CAR T-cell therapy for diseases like multiple myeloma, a blood cancer, on off-the-shelf immune cell therapies that'll be ready for when patients need them, especially for those patients who may not be healthy enough to have their own cells engineered, on editing blood stem cells to correct genetic disorders in children with the potential to be truly curative. This is a revolution in the way medicines are manufactured and people are treated, and we're at the forefront of it. Every time we work on a new living medicine here, it strengthens this local ecosystem. That matters because the local manufacturing isn't just about the scientific progress. It's also about access, cost, and developing promising therapies that can realistically reach more patients. With this infrastructure and innovation, we're setting our sights on even bigger goals. Today, the majority of cell therapies still rely on taking cells out of the patient's body, engineering them in the lab, and giving them back to the patient. But what we're working on now is helping build towards a future where we'll be able to give those instructions directly to the patient so the engineering can happen within the patient. When that becomes a reality, it'll be transformative. It'll make these therapies simpler to make, easier to scale, more affordable, and much more accessible to the patients who need them. Sometimes it still sounds like science fiction, even to me, but it's not. It's already happening and it's becoming real. And in that future, local manufacturing will still be just as important. The delivery system, the materials, the careful checks on quality all still have to be designed, built, tested, and made safe for real people. What we're building is not just about now, it's about a foundation for the next generation of therapies as well. Here in Calgary, we're not just getting ready for the future of medicine, we're helping build it. Thank you.
Thank you so much. I think even as someone who does a lot of writing in this space, it is astonishing to me the kind of work that's being done here. And I think we'll go from this sort of extraordinary edge of science to looking at something that's, I think, is much more elemental, and that's the relationship between patients and researchers. And our next speaker is actually two speakers, and because one of them comes from broadcast, you're going to learn about them through a video before they get up here to present. But I'm going to introduce them first. Camry Imlach is a master's student in immunology at the Arnie Charbonneau Cancer Institute under the supervision of Doug Mahoney. She is focused on developing strategies to treat acute myeloid leukemia, AML. You might already know David Spence. If you're like thinking, gosh, I know that name, you will know as soon as he stands up and you hear him speak. He's a retired Calgary broadcaster and meteorologist, and he was telling me very depressing things about the weather, so I'm glad you're spending your last sunny night here with us. He is a prostate cancer survivor, and he spent much of the last decade volunteering as a patient and family advisor in the provincial cancer care system. More recently, he's been working very closely with researchers, and it's quite a beautiful thing to see that relationship up close. So we'll start with the video, and then they'll come up to chat with you.
By the morning, they had diagnosed him with leukemia. You know that lots of people don't survive it. You know that it's scary. You know that it's cancer. You know that your life is going to change forever from that day.
My name is Camry Imlach. I'm a master's student at the University of Calgary, and I am a cancer researcher. AML is a blood cancer, and it's one of the most fatal types of blood cancer. So currently it has a really poor prognosis. Working on CAR T-cell therapy for years and not meeting a single person who had received it doesn't make all that much sense, really, when you think about it. Before this, I had never heard of patient-oriented research or patient engagement.
Calgary saw some cloud today. My name is David Spence. I did weather on television in Calgary for almost 40 years, and through that, I think I gained some experience in conveying scientific information to the general public. I was diagnosed with prostate cancer in 2012 and treated in 2013. That was many years ago now, but ever since then, I've wanted to give back.
Poured into this mold in here. David set up a Zoom call between me and a man who CAR T-cell therapy saved his life.
You know, a light bulb has gone on here and something has really clicked. They made a connection, a very good connection, an amazing connection. And, you know, she told me afterward that conversation changed everything for her. It was one of the greatest days she had in her life. And I thought, that's excellent.
You can see the colonies.
Callan and Janelle, just the way the conversations went and the way that Camry realized what her research will one day mean for children like Callan.
They told us he had a 60 to 70% chance of survival. Devastation. He ended up doing 5 rounds of chemo, and because of the kind of cancer he had, he was considered high risk. So the chemo that they have, they get chemo and then it knocks down their blood counts and they have to stay in the hospital until their blood counts come back. So we ended up spending most of 5 months in the hospital.
Every time you get a needle or every time you kind of suppress something, or each day spent in the hospital, each night, then you get a bead.
So there's a different bead for each thing. So one for chemo, one for a night in the hospital, one for a blood test.
And then at the end of your treatment, you get it, and it's full of colors and full of beads. I just kind of hope that we find some breakthroughs, of course. More easier treatments to go through.
Definitely thinking about the impact that this therapy could have helps push me through more difficult times in my research. My research is all about making CAR T cells for AML. The goal of research, I think it does take on a new meaning when you work with patients. It changes the way that you look at your research and that you conduct your research, and definitely the motivation you have for it. My work has purpose.
If researchers asked you for your help, would you be willing to help?
Yes, definitely.
The best thing about research is that it changes what's possible, and it gives hope.
Research changes what's possible. It gives hope.
And with hope, there's impact. For nearly 40 years, I translated complex science for a television audience. That science was whether this is cancer. In 2012, I was diagnosed with cancer. Fortunately, it was a highly treatable form of prostate cancer, for which I received treatment a few months later in 2013. That was a long time ago, but a cancer diagnosis changes your perspective forever. It creates a powerful drive to make life better for others who face cancer. Becoming a patient partner in cancer research is one way I give back to those who gave my life back to me. Traditionally, doctors and oncologists see patients. Researchers don't. That's changing. And patient-informed research gives scientists a new, valuable, and motivating perspective to the work they do. Patient engagement takes many forms, and you may not be familiar with it. In our co-mentorship program, patient partners are paired with researchers. I'm attached to Camry, but I'm not a member of the research team. I bring lived experience personally to the research partner, to Camry. In other settings, patients may serve as team members or as advisors, but the goal is always the same: to ensure research reflects real lives and not just data. As a patient partner, I bring lived experience both in life and in cancer. It keeps the human element at the forefront of the researcher's work. And drawing on my background in communication, I've been able to help Camry and other brilliant young researchers translate their work into plain language and strengthen their public presentation skills. Importantly, this will help them in their careers. More importantly, it will lead to better research, and most importantly, provide better outcomes for patients.
From a young age, I had family members who were affected by cancer. I had a little cousin who passed away from a brain tumor at age 5, another cousin who passed away from cancer in his 30s, and more recently, my grandpa beat bladder cancer, and my other grandpa, who is here tonight, beat prostate cancer. This is what drew me to a Bachelor of Medical Science and now a graduate degree doing cancer research. 2 years ago, I joined this lab, and I was introduced to CAR T cell therapy. I was told about this amazing treatment that was helping cure people with cancer who had run out of other options. And that is what made me want to be a part of this work. But as an actual researcher, most of my time is spent in the lab, far from any patients. I do lots of experiments, I read papers, I think all about science, and the real effect that my work might one day have on patients can feel very distant. Meeting David in the fall, and then being connected through him to other patients has helped change that. At the beginning of this program, I didn't know what to expect because I didn't know how patients could have a role in research besides being the subject of it, or maybe an extra set of hands to help with my experiments. But now, after meeting and learning from patients, I understand much more clearly just how much their voices matter.
What has made our partnership work so well begins with Camry herself. She embraced the concept of patient engagement right from the start. The co-mentorship program is not mandatory. Camry gets no course credit for this. She enrolled because she wanted to be there, not because she had to. Now, my experience is not just my own. My father had colorectal cancer, and that is what led me to team up with a colorectal cancer researcher in the co-mentorship program last year. My mother had leukemia, which brought me to Camry this year. So in doing this, I am honoring my parents. One of the most meaningful moments from my partnership last year with the PhD candidate studying colorectal cancer was connecting him with a colorectal cancer patient. Until then, he'd never met one. That Zoom call changed his perspective, and from that moment on, he put patients at the forefront of his work. So I brought that same approach to my partnership with Camry. My cancer was prostate, and I was treated with radiation. Camry is studying CAR-T immunotherapy for AML. My cancer and her research are not a perfect match, not at all. So I set to find out CAR-T and AML patients. And through some of the wonderful people at the Leukemia and Lymphoma Society of Canada, I was able to connect Camry with a patient who had undergone CAR-T therapy. I also introduced her to a caregiver whose father had undergone CAR-T. The impact of those conversations on Camry was transformative. Camry was no longer a researcher peering through a microscope. She was peering at real life and at the possibilities.
Even though I study CAR T-cell therapy, I had never actually met anyone who had received this treatment. So when David suggested connecting me with a CAR T-cell patient, I was incredibly excited and honestly a little bit nervous. I had spent so much time learning about CAR-T from a scientific perspective, but I had never heard firsthand from someone who had actually gone through it. This was my chance to find out whether the lived experience matched the hope that I had placed in it. And if anything, that conversation made the promise of CAR-T cell therapy feel much more real. This patient had relapsed diffuse large B-cell lymphoma and was given a 33% chance of survival with chemotherapy and a 42% chance of survival with CAR-T cell therapy, which was very new at the time. For him, that was a significant difference. He ended up being one of the first people in Canada to receive CAR-T through a clinical trial. And he went through some of the more severe and painful side effects associated with this therapy. But that is what made it so very powerful to hear that he was so grateful for it. I remember he said something along the lines of, "If you're looking for someone against CAR-T, you won't find that here. I am a huge advocate for it."
On the Zoom call, the patient was in his home, in front of a wall, on which he painted a Bob Marley quote, a quote that captures everything patients endure. "You never know how strong you are until being strong is the only choice you have." The strength of caregivers is also tested when a loved one has a cancer diagnosis. We also had a conversation with a woman whose father underwent CAR-T therapy, and she was his caregiver.
The caregiver that we spoke to actually had a much better experience. She told me her father found CAR-T to be far more tolerable and less intensive than chemotherapy. What really stood out to me is that even with very different experiences, they were just so thankful for it. And gaining that personal side to my work felt so encouraging. I remember ending those conversations and just thinking, wow. But those conversations were also really emotional. I heard people talk about what they went through, what it was like for their families, and the painful parts of both the cancer and the treatments. Having those conversations taught me things that I never could have learned from a textbook or the lab. It gave me a deeper connection to my work.
The research continuum has both a starting line and a finish line. Camry, as a first-year's master's student, is at the starting line. Callan, a 12-year-old AML survivor who you met in the video, enjoyed the victory of crossing the finish line.
Getting to know Callan and Janelle was another experience that was very meaningful. I was introduced to a real person, a child who had AML. And one of the biggest things I realized was that it changed the way I think about impact. In research, we use survival statistics to explain why our work matters. In Canada, for AML, the 5-year survival rate is only 26%. I use that statistic all the time. The pediatric cohort of AML is considered to have a better survival rate of around 60 to 70%. On paper, that can start to look more reassuring, but when I got to know Callan and Janelle, that perspective completely changed. Imagine being told that your child has a 60 to 70% chance of surviving. That's absolutely terrifying. When you put a real child and a real family behind those numbers, the need for better therapies feels far more urgent. And so having this patient engagement and connecting research to real lives has changed the way that I see the work I do every day. And all of this was really made possible because of the mentorship that I received from David. He's helped me grow in many professional ways, my communication skills, networking, outreach. But beyond that, he helped make me a better researcher because he understood what would be valuable for me before I even did. And so I'm really grateful we were paired together because of the way he has supported not only my research career, but things that have shaped who I am outside of the lab. Even though our formal partnership is technically over, I have a feeling that his mentorship will be continuing for a long time. Can't get rid of me anytime soon.
You got that right. There comes a point when you recognize the people who've truly left their mark on you, the ones who shape not just where you've been, but who you are becoming. That list includes Susan, my wife of almost 34 years, who's sitting right there. A high school teacher, Milt Wright, who helped me launch my own career, and a boss in Kamloops, Jim Harrison, who 46 years ago taught me more in 5 months than I'd learned in all my years before, and probably since. And Camry Imlach. She's on my list. And let me tell you why she is on my list. She's on my list because it's not only because of the remarkable person that she is, it's because of what she represents, the very best among us. When I was treated for cancer, those treatments that I received were developed 30 to 40 years earlier, imagined by someone I will never know, never meet, and never be able to thank. And that humbles me. But it also sharpens my appreciation for the people who are doing that work today, right now. And Camry is working today on research that may help save lives decades into the future. People she will never meet and never know. As someone living well after cancer, meeting Camry and getting to know her and working with her has been so fulfilling. In the magnificent career she has ahead of her, she will encounter frustrations as well as successes. There will be challenges, she'll be underpaid, and will probably have to fight for funding. But she knows it's worth it. And as she said in the video, her work has purpose. Camry, you spoke today about how meaningful it was for you to meet CAR-T and AML patients, and I want you to know it's been equally meaningful for me to work with you and to get to know you and your remarkable colleagues. For patients across every cancer diagnosis, researchers like you are the quiet heroes, working behind the scenes, often unseen, and quite literally, the people many of us could not live without. So no, you can't get rid of me either. We're a team through and through. Again, I say this as someone living well after cancer, but also as a friend. You are making a difference, a real one. And from my heart, and on behalf of the cancer patients of the future, for what you do and for who you are, thank you.
And I would also like to thank all of the patients who've taught me and everyone who helped with the video, especially the Trongs, Janelle and Callan.
All of you were so great. You two in particular are the cutest. So I have spent so much time with you over the last few weeks, and I still get emotional watching the two of you and the way you connect. So thank you for that. I want to start with a question for everybody, because I think what we've been looking at today is this pathway between, the idea and then a therapy getting to a patient, as David put it, like the start line and then the finish line. And we have seen all these different challenges in these gaps along that pathway. And I wanted to hear from each of you, what do you think is the biggest challenge from your perspective to be overcome in that sort of space between discovery and getting a therapy to a patient?
I'll go first.
Unfortunately, you're closest to me, sir.
So I think from the clinician side, we see that there's this great amount of research. The science is there. There's no problem with science. The biomanufacturing is getting honed down. It's getting it from that place into the clinic that is our biggest challenge. Some of that is regulatory challenges. Some of it is how do we get some of these things that have already been proven? So, for example, ADA skid gene therapy is a great example. As I mentioned, over 100 patients have been treated. Those have almost all been on clinical trials. So that therapy has been really difficult to commercialize. They're very expensive. They're not like a typical treatment, like a blood pressure medication that is made in a factory for hundreds of thousands of patients. We're making something that's living cells, and it's not profitable to commercialize. So that's one of our biggest barriers. There's a number of different gene therapies that have gotten through the clinical trials, but our barrier is how do we get it into standard practice? Because our traditional pharmaceutical commercialization pathway is not a good fit. So we need some creativity to figure out how to get through that.
I think the easy answer would always be funding. But, I think it's been really building these ecosystems that, connect the scientists, the patients, the clinicians. So there's the full pipeline. I think for so long, everything was very siloed. Scientists were working alone on their research projects with no connection to patients, no connection to the clinicians. And I think building that and what we're building here, is really paves that path. So, what is the clinical problem can help inform the research questions. What kind of therapies, and experiences do the patient want? All of that helps inform and better drive, ideas to actually have outcomes for patients.
I don't know how much I have to add. I'm pretty new, but I always thought that there's some difficulty taking the preclinical data and sort of proving that it's enough for a clinical trial and proving that it's safe enough, and getting all of that done. And so I see that as a challenge.
I think that the safety protocols that we have in Canada can act as a barrier in many ways. Of course, it's important that any clinical trials be conducted under the safest of conditions. But from what I understand, a lot of things don't get to clinical trial in Canada very fast compared to other countries because we have such a strict safety protocol. And if there is a way to smooth that out, there is a way to not reduce the safety, but expedite the system so that we could get to clinical trial faster.
Sorry, I had a follow-up question that just went clear out of my head. That was not what I expected you to say, and that was great.
What did you think I was going to say?
Okay, well, let's go to my list because I can't rely on my brain today. I know what it was. Not last. How do we make Canada not last? Which is following on what David said. Is it the same thing we're already doing, but expediting it? Or is it some sort of creative framework that we have not thought of yet that gets these therapies to patients faster?
So we can go in order again. So I think that's where we need to start talking about investment. So I think the amount of funding that we get pales in comparison to, for example, the United States. So some of my research is actually heavily supported by the California Institute of Regenerative Medicine, who that has been a big priority in California for research, is to funnel it into things like gene therapy and cellular therapies. So, for example, our CD3 Delta Skid product, we're getting almost $6 million from California. Here, I got $2 million. So I think that is a huge difference. There has been a national rare blood disorder strategy. That money, a lot of it is being funneled into trying to give patients drugs that aren't covered, but more of it needs to go into investing and getting these things into clinical trials and developing them in Canada.
Yeah, and I think it's really interesting what David said, because I think that's probably the typical patient response, is we need to be more aggressive with it. And I think the scientist and clinician response is actually being more conservative. And there's probably a blend of where we want to be with that, and working with, the different advocacy groups out there that are, pushing for regulatory reform to try to make those things happen, with the patient voice there, because, it's been a change for me in hearing patient stories, is that they want, maybe more risk than some of the clinicians and scientists are willing to take to get these therapies ahead. So,
I mean, is there a way to take it on an individual patient basis? If you have a patient who's run out of options and there's this clinical trial that has not yet quite received approval, is there a way to expedite it for that patient who literally has nothing to lose by trying this out?
I'd say partly yes, and that's, the two patients I spoke to that have been treated here in Calgary with the CAR T-cell product, they were in kind of that method. It wasn't a full phase 1 clinic trial. They were these single patient studies. There's still, more or less the same rigor that goes into it, but it's a way to get it to an individual patient when we haven't had a larger trial up and running yet.
Right.
Well, I have a question that's for you to start. And so one of the things that I have learned a lot about watching, helping with the video and chatting with you over the last while is how much you work like in this lab where you don't really get to interact with patients unless you seek out those relationships. And I want to know, like, to what extent do you feel that patients should be like really involved in the research process early on? Like, where should this interaction begin between researchers and patients and their families?
Yeah, it's a good question. And actually, David and I talked a lot about that when he first became my partner, because I was just starting my master's. It was September of my first year of my master's, my first month. And so we talked about how nice it was that I could have this perspective from the very beginning, and that I could start my project, like before I'd even begun really the research for it, with the patient voice in mind while I'm trying to develop a new therapy for AML. And then meeting like Callan and meeting CAR T-cell patients, I can have that inform the plan for this therapy. And then also along the way, it's definitely important to keep yourself in check and to always continually, I think, include the patient voice in your research. But I think especially having trainees work with patient partners and not just scientists, because it's a new thing almost that's starting to happen where the patient voice is getting included more and more. And so it's good to have the young trainees starting who will grow up to be the scientists.
David, do you have anything to add?
I think it means everything that patient partners get involved right from the start. There have been cases where a project will be midway through and somebody goes, hey, I need a patient partner to get some grant money or something like that. So they recruit us and we're there to tick a box. That's not genuine patient partnership. But with this program, I'm involved in a couple of other projects as well. One is also at the University of Calgary involving nature-based therapy. And I was brought in fairly early from the start. And that makes it meaningful to me to join that team as a patient partner, a patient advisor, and work with them from start to finish. Who knows where finish is? It could be a few years from now, but it's underway. I recently joined one in Vancouver at University of British Columbia, which is a prostate cancer project, which I really like because that's my cancer. And again, it's at the very start. In fact, the project hasn't even begun. They're applying for grant funding, and I'm quite proud that my name is on the grant application as a patient advisor. So that's right from the very start, and I'm quite excited about what lies ahead with that one. So it becomes very meaningful for the patient to be involved right from the beginning.
I'm aware there is this tension in terms of how we fund things, that if we look too much in sort of things that are about to be like applied in a clinical setting, we might not be funding the basic science enough. And so how do we deal with that?
It's a challenge. We're, up here, I think most of what we're talking about today is very translational, patient focus. But all of those technologies come from obscure scientists being curious. The technologies Nikki was speaking of were studying bacteria for no purpose other than being interested in how bacteria fight infections. So I think it's important that, you let scientists be curious, because that's what fuels these, endeavors decades later, much like David speaking of his treatment being, designed 40 years before he got it. The stuff being discovered now in worms, might be the next breakthrough in years to come, but we just don't know.
Yeah.
I would definitely concur with that.
I think there's sometimes a focus on applied science because you can see the end and the impact and what that's going to do. But as you said, the applied science, like all the CRISPR and base editing and prime editing, wouldn't have happened if that basic science in that bacteria, in that little obscure lab, hadn't happened. So you have to keep letting the scientists and keep funding them to do these things that might not, you might not know what the end of that tunnel looks like. The end of that tunnel might be something that's going to revolutionize care.
Right.
So you have to keep going.
I think in this environment, sometimes that can get challenging because there's limited funds, and there is this expectation that especially public dollars are going to be going towards things where there is like a very high outcome that it's going to help somebody. So I don't know.
We have to protect some of those funds for the basic science.
Yeah. I want to put all this in perspective, because we talk about, CAR T, and we talk about gene editing, and we hear about patients who are getting these therapies. We hear little bits. Can you tell me who is getting these therapies right now? And are you comfortable with that? Should it be more? Are we right where we should be?
For the gene editing, I'll talk about that, and then maybe you can talk about the CAR Ts.
Yeah.
So, for example, for patients in Canada, the patients who are getting gene editing have to leave the country to get it. So we, for example, have had 3 children in Alberta who have gone to the United States for clinical trials to get gene editing. I included Jacob, one of the ones on one of my slides. It's really a very small percentage of the population. I think the other challenge is sometimes it's the patients who have the loudest voice behind them. So there are lots of families who are philanthropic who have raised lots of funds to develop these therapies for their children. But that leaves the families that don't have those supports without that therapy. So right now it's a little bit who's got the loudest voice and who has the resources to develop those therapies. We really need to try and make sure that it's equal. We are currently developing a therapy for a couple of underserved groups. So we've taken that on ourselves. It hasn't been because the patients have been making lots of noise. So I think we have to make sure that we balance that along. But I think we also have to bring it into Canada and not keep sending our patients other places for these things.
And before we bounce to Kyle, how and when do we get there?
So we are planning to start treating patients here in the next 1 to 2 years. The one thing that even further along the way, so one of the diseases we're targeting, there are patients in other places of the world. I'm looking forward to the day that we bring American patients to Canada for treatment.
Okay.
So we are on that path.
Nice.
Yeah.
Kyle.
Yeah, I think the CAR T cell world's maybe a little bit ahead in patients getting access in Canada. We treat patients with CAR T cells in Canada. There's 6, I believe, approved in Canada as, standard of care. But they're all manufactured in the US. So the patient's cells are collected here, sent to the US for the product to be manufactured and it's shipped back. I think the shift we really want to have is to be making those products here for patients here. And that's, work we're doing here, but also other centers across the country are all collectively trying to, have these made in Canada therapies to, reduce the cost and then increase the access for Canadian patients.
Anything to add? No, it is unfair to ask you. I do want to hear from everyone. If you're looking ahead, 5 years, what is the one thing that you expect to come out of what we have talked about tonight that you think will be a reality? Maybe American patients coming to Canada?
That would be amazing. I think the other piece from nights like tonight, I think getting more awareness amongst non-scientists as to what's going on and how important it is, and advocating for changing our system, things like the regulatory pieces. That's where we need the voices to help tell our government, for example, these are the priorities and this is where the investment needs to go, and supporting that. And I think the other big shift that I'd love to see in the next 5 years is, and I think this is an opportunity we have in Canada with our public health system, is I think we have an opportunity to move away from this big pharma, this is the commercial pathway that these things need to go down. I think we can start developing things in Calgary, put it through clinical trials, and I think we can start making it not commercialized for a fraction of the cost here. So that's what I would like to see. Okay.
Yeah, I completely agree with Nikki. I think that last part is huge, is figuring out the mechanism so we can get really reimbursed, just to cover the costs that we need to do to provide these therapies more widely. But I think the big change will come in some cases is, both Nikki and I touched on it, is, the engineering happening in the patients, as opposed to having to take cells out, engineering them in the lab, this, in-person engineering. I think that's going to be really the next revolution and stuff we're, have an eye towards to try to be at the front of.
Yeah, I'll talk a little bit more from David and I's role point of view. And I hope that in 5 years, and I think in 5 years, there's going to be less of this divide between researchers and patients. Like programs like the co-mentorship program is already spreading more, growing. There's interest in it. And so I think that will be more common. And I think that lots of trainees will realize that they have this opportunity, or that they can in some ways seek it out themselves. And also just nights like tonight where research can become less foreign for non-scientists as a whole, and it won't be so many people wondering what the new treatments might be or what's happening. And so that's something I look forward to.
So well said. In 5 years, Camry will cure cancer. 5. Realistically. I think one thing that we need to do is encourage our researchers, encourage our trainees, encourage our students. The work that people like Camry are doing can be tedious. It can be discouraging from time to time when your experiments, one after the other, don't work out the way you want it to be. And though you learn from a failed experiment just as you do from a successful experiment, I think we have to provide our researchers with the motivation to keep on going. And patient engagement, I think, is an amazing way to do that. I've seen it myself with at least 2 researchers. And I think if we can keep these people motivated, it's just going to make it so much faster that we get to the point where we really do come up with effective, inexpensive, and hopefully one day commonly used treatments for the worst of these cancer diseases.
Thank you. So we have some people with microphones somewhere. I'm a bit blind, but I think we have them. And so if you have a question, just put your hand in the air and wave it very frantically, because we're being blinded up here, and that's okay. But just make sure the people with the microphones can see you. No questions. We have some down here. We might have some in the back. I just might not be able to see you.
It's impossible to see, isn't it? It's all black up there.
Okay, hi everyone. First of all, thank you to all the speakers. It's been so great to listen to all of you share your stories and your research. David and Camry, this question is kind of for you guys. I hear you guys talk about, or David, you specifically talking about wanting to expedite the process of getting clinical trials to patients. And when I think about that, I think about it from the clinician perspective of risk and informed consent. Yeah. And I'm just wondering if you think there's like a role for the co-mentorship program and more patient, like researcher trust to expedite that process. Do you think if there's more trust between the general public and researchers, those things could get expedited?
I think there are probably ways to get to that point. I don't know. The co-mentorship program as it exists is for preclinical research. So we're not at the point of patient care. We're not at the point of clinical trials or anything like that, which is not to say a co-mentorship program can't be developed for that stage, but it's not there yet. So I don't think, does that make sense that it wouldn't apply to the co-mentorship program that we have now? But your point is really, really good. And, I was at a conference in Vancouver about a month ago now, I guess. Is that a month ago? Something like that. And we had a discussion in a small group about the fact that it takes, on average, about 2 years longer for something to get to clinical trial in Canada than it does in other countries. And there was a lady there who had worked for Health Canada, and she defended this. She defended the safety and whatnot. And a lot of people in the room were going, yeah, but, in this country, they get to clinical trial a whole lot faster, and people are benefiting from that. So I really think there has to be a way to expedite things. The co-mentorship program, I'm not sure as it is now is the way to do it, but something like it, let's develop it. Let's do it.
Yeah, I think more engagement so there's more trust in research can be really important at the preclinical stage, and then it could also be really important moving more into clinical stages, because the trust can be a barrier. For example, getting samples to work on sometimes it can be difficult to get consent for and stuff like that. So I think having patient engagement and building that trust could work really well there.
With one of our projects, we work with an underserved population that has a lot of barriers for research. So we have actually developed a patient engagement advisory committee, and part of it is working on some of those issues with how do we engage with this population that maybe is not as educated about science, but also has a lot of hesitancy and lack of trust. How do we build that when we actually want to be doing some of these really novel therapies in this patient population? And I think that's actually been a really remarkable eye-opening experience to some of the things that are important to them and that we will keep in mind with our clinical trial.
Kyle, I think about your work because so much of what you talk about is sort of bench to bedside and then back again. And from the biomanufacturing perspective, like how much do you work with patients, or would you like to work with patients?
I'd say we interact with them a lot through the scientists, or through the clinical side. Less been directly in our space, but there would be no reason not to. But a lot's been through the scientists and those projects and how they've developed. And sometimes it's we're just kind of trying to put together the nuts and bolts to make it a product, and then it's onto the clinical side. So it's partly there, but maybe not to the same extent as a partnership of David and Camry.
All right, more questions. People, don't be shy. I could hear footsteps, so I figured we were going somewhere.
Is there any research being done as far as the best preventative and treatment measures in terms of lifestyle for preventing or treating cancer? And as a side to David, how is your cheesy supply doing?
I have a house full of cheesies. I do.
Maybe to the prevention, this group here, that's not been our focus, but there has been, previous cracking the cancer code discussions and whole, departments at the university that are really focused on prevention, lifestyle, and even outcomes on the other side of it. So, this probably isn't the crowd to address that question perfectly, but it's a significant avenue of research at the university here, at least.
Well, I have a big regulatory geek question, so step up. I am very interested in this discussion about what can we do to set up a framework that would, if not speed the approval of these, just ensure that there were needless barriers in the way. And one of the ideas I've heard suggested is that gene editing is, could be approved as a kind of molecular surgery. And so if we have if someone is developing a new kind of operation for something, it doesn't have to go through all the regulatory steps of a new medication. Is that, you think, the way that this will go?
I think that's one of the things we're pushing for. Actually, you had the best term for it, the technology platform. So, which would be sort of similar to your molecular surgery. Right now, to get any of these novel therapies through, you have to go through this regulatory process, and there's all of these things you have to do to prove it works, to prove it's safe, prove it's like the product is good. It's all designed off of your typical small molecule drugs. So we're trying to fit these things into this, like the square peg into a round hole. So we're actually proposing to Health Canada that, this is different and needs to be treated differently. Can we create like a platform for these cellular therapies where it's a little bit, okay, these are the things we have to do. For gene editing, for example, if we've proven a base editor it works well and is safe for one disease or for one variant in one gene, can we skip that step and just prove some of the other things that need to be done for the next therapy that's using that base editor? There are some things that we've had to do previously, like mouse models and things that were like, is that actually adding any value to the safety? We've already proven it kind of works. So we're asking the regulators, okay, let's pull it back and have a look at this, and how can we make this a platform so that we can reduce the amount of work that we have to do for each of these things. Okay. So I think that's one direction we really want to go. And the FDA is also looking at that route. So I think they're starting to see that this is a problem.
Is there anywhere in the world that you think has figured this out?
There are some other countries. So, like, for example, Australia, they have a different bit of a pathway for some of these one-off things. So, for example, the G card that you talked about here had to go through this single patient study. We had to put together all of this stuff for Health Canada, prove all these things. In Australia, the regulators actually rely more on the team using it to decide that it's safe. So they wouldn't have had to create the single patient study that would have had to been done here. They would have said, okay, we see what you're doing. It looks like it's safe. Go ahead. But you don't have to do all these other rigorous things that didn't actually add anything to the safety. It just added more work and more expense. So Australia, for example, has a good system. And some of the European countries also have these hospital exemption pathways where you can get these things through much faster. So we're actually advocating with Health Canada to try and change the system.
Well, I'm going to bring it back to the two of you and ask a question I thought for sure someone was going to ask here. And that's you have this incredibly special bond, and that really came through tonight. I think a lot of people will want to be part of something like that. And so if there is a patient or a family that wants to get involved in co-mentorship or in patient engagement research, what should they do?
For the co-mentorship program, you can send an email to charbonneau@ucalgary.ca. Then you can get some information from there. But this isn't the only patient partnership program by any means in existence. Canadian Cancer Society, Alberta Cancer Foundation, they have resources to hook you up. You can also try organizations that involve specific cancers like leukemia and lymphoma, for example. They're fantastic people. They help set us up with the interviews for Camry. Most of the advocacy groups and organizations have patient partnership programs. In my case, I started over with AHS as a patient and family advisor. That is another terrific place to get into the program as well. They have so much to offer. When you start out, you might be, I don't know, filling out a survey or two for the first few months. But then as you get more into it, you're going to get invitations to committees, and you're going to get invitations to focus groups and that sort of thing. You're going to be part of discussions and panels where your voice actually does matter. And I would encourage anybody who's interested in that maybe to try what was AHS, now Cancer Care Alberta, or Primary Care Alberta, Acute Care Alberta. All those organizations have patient and family advisory groups. And if you're interested in that, that's definitely a great starting point.
Yeah, and that goes for trainees too and grad students in Charbonneau if you're interested in getting a David.
I'm taken.
Well, I don't think we have any other questions, and so we might just end on time for. Oh, there. Okay, sorry.
I assume that all of these gene therapy areas are done out of universities. How many other universities in Canada are doing it?
Great question. I would say in the blood and immune disorder side of things, and with the gene editing, with the base editing and the prime editing, for example, we're leading the charge. There's some work being done in like some of these areas at Saint Justine in Montreal, a little bit at Sick Kids, not so much at UBC, but honestly, I think we actually are leading the charge. We have the biomanufacturing is also, I'll let you speak to that piece of it, but we are really building here.
Yeah, I think Calgary's very much a leader. A lot of the other big centers, Toronto, Vancouver, they have programs like this, but a lot of those, at least in the biomanufacturing case, those have come out of the standard of care side, and they haven't really been purpose-built for these novel therapies. And I think that's something we've done uniquely here is everything is geared towards trying to move things to patients, learn and iterate. And I think that's really going to put us at the forefront in Canada, in the cancer space.
Okay, help me out. I can't see at all. Is there anyone else?
Sorry, I have one more question. So it sounds like you all need more money for your research. And what is the mechanism by which that would happen? I mean, yes, the government, but what else?
So the grant funding through the federal government, provincial government is part of it, but the biggest part is you'll see both of the the Charbonneau and the Riddell Center are named. A lot of it's philanthropy is the driving force, and that's been something I think that's what's allowed us to be so unique here in Calgary has been the philanthropic community and the generosity there, their engagement in what we're doing and what we're building. So that's really allowed us, I think, to leap forward with what we're doing.
We were lucky with the cellular therapy program to recently get a huge investment from the Children's Hospital Foundation of close to $9 million, which is fantastic. So that's an avenue that people can go through. And then the Cummings School of Medicine is also through their donation pathways is where a lot of the philanthropy comes through as well.
Well, I think I'm getting the sign that we are done. So I have a number of thank yous, and they're sincere, even if I can't make eye contact with you. But I want to start with Janelle and Callan, who are here tonight. I don't know if they want to stand up, but one of we didn't we were trying to do this video quite tight, and one of the things that didn't come through was the fact that Callan had a stem cell transplant, and I know it is very important to them that you hear that it's important to donate, that they want stem cell donors, and so please take that message home with you. I also want to tell you that Callan is a black belt, and I'm a black belt in what? Taekwondo. And we couldn't get that into the video either, but that's pretty amazing. I want to thank the library for hosting us and the caterers and for their fantastic cookies. I want to thank all of you who just delivered beautiful talks and worked so hard and just did a perfect job of translating between the very specialized things you do and making sure that we understand it. And to the two of you for letting us be a part of this fun relationship that you have. To the communications team at the Charbonneau Cancer Institute and the Charbonneau Trainee Association. And I want to say thank you to all of you. You're here. It's the nicest day we've had in a long time. It's the last nice day we're going to have in a while. This really makes a difference to have people in the space talking about these kinds of things. And we're so grateful for your presence. So thank you for coming. Please come again. Thank you to everyone here, and get home safely.
Thank you for moderating.